When we have genetic variation in a population, we can say that changes have been made to the gene pool. And it literally is spelled pool, like a swimming pool. So you can imagine it as like the biggest summer barbecue you've ever imagined. And this is an in-ground pool because we're bougie. Okay, here's our little house. That just goes to show you how large the gene pool is. But we're having our summer barbecue, hanging out, partying, and the whole population is in the pool. It's hot. No one wants to just be sitting outside. Everybody is in the pool. So the gene pool here is all of the genetic information within the population. So when we're talking about this, we're talking about genes, we're talking about alleles, we're talking about nucleotides. And for organisms that don't have DNA, we're talking about their RNA too. So now that we know what a gene pool is, we now have to ask ourselves the question, how does a gene pool of a population change? Gene pools can change in two ways. It can be reduced. So you're reducing the genetic diversity present in that gene pool. But gene pools can also be expanded. And it's expanded because we're increasing genetic diversity. There's more alleles, more genes, more nucleotide sequences that are in this gene pool that are influencing it. So we're going to go through each of these. We're going to go through two ways that a gene pool can be reduced. The first is called the founder effect. And the second is called genetic drift. Then we're going to talk about the ways that gene pools are expanded. That can be through sexual reproduction. And the other example that we're going to use for this video is gene flow. Another way that we can expand our gene pool is through mutations in a population. But because we talked about mutations so much in our genetics unit, we are not going to sketch this out.